Hello viewers, welcome to Sleepy Mind Channel. Tonight, we'll be exploring aging. Imagine walking into a high-tech medical clinic, pulling out a small sterile swab, and scraping the inside of your cheek. You hand it to a technician who drops it into a sequencing machine. You are 35 years old according to your birth certificate, your driver's license, and the candles on your last birthday cake. But an hour later, the computer screen flashes a single, chilling number, 44. Your birth certificate is a legal fiction. Deep inside your cells, your biological gears have been turning at double speed. You are nearly a decade closer to your grave than the calendar claims, tracked by an invisible molecular clock that has been counting every stressful deadline, sleepless night, and toxic environment you have ever endured. This is the reality of the Horvath epigenetic clock, a groundbreaking discovery by biostatistician Steve Horvath that revolutionized how we understand the physics of aging. For centuries, humanity defined aging by the steady, unyielding ticking of chronological time. We assumed that everyone decays at the exact same rate because we all spin around the sun at the same speed. But biology does not care about astronomy. The epigenetic clock tracks aging by measuring DNA methylation, a process where tiny chemical tags called methyl groups attach to your genetic code. Think of your DNA as a massive piano keyboard containing 20,000 genes. When you are born, your songs are crisp, clean, and perfectly tuned. But as you live, the environment presses down on the keys. Stress, poor diet, trauma, and pollution leave sticky chemical residues on the keyboard, permanently silencing certain notes and forcing others to play out of tune. By analyzing roughly 353 specific methylation sites across your genome, algorithms can calculate your true biological age with terrifying accuracy. The human impact of this science is profound. It means that age is not a passive slide down a timeline. It is a dynamic, recorded ledger of your life's choices and struggles. Two people can be born on the exact same day in the exact same hospital, yet one person's molecular clock can run 50% faster due to systemic poverty, chronic anxiety, or genetic predisposition. The epigenetic clock strips away the vanity of cosmetics and lifestyle trends, looking past your smooth skin or dyed hair to read the raw, unvarnished truth written in the chemical shadows of your chromosomes. It proves that our bodies are ultimate historians, keeping a flawless, cellular diary of every wound we fail to heal. Right now, deep inside your tissues, a microscopic horror movie is playing out. Millions of your cells have reached a terrifying existential crossroads. They have become too damaged to divide, but they refuse to die. Instead of committing cellular suicide like healthy components should, they mutate into immortal, deformed entities known as senescent cells. They are biological zombies. They sit trapped inside your muscles, your brain, and your heart, completely unresponsive to the normal laws of biology, actively secreting a toxic chemical soup that corrupts, poisons, and degrades every healthy young cell surrounding them, dragging your entire body down into systemic decay. Cellular senescence was first discovered by Leonard Hayflick in 1961 when he realized that human cells can only divide a finite number of times, a boundary now known as the Hayflick limit. When a cell approaches this limit, or when its DNA suffers catastrophic damage from ultraviolet light, toxins, or metabolic stress, it undergoes a radical transformation. To prevent itself from turning into a runaway cancerous tumor, the cell flips an emergency kill switch. It stops dividing forever. However, instead of quietly dissolving and allowing the immune system to clear away its debris, the senescent cell enters a hyperinflammatory state known as the senescence-associated secretory phenotype, SASP. It begins pumping out a highly destructive cocktail of cytokines, chemokines, and matrix metalloproteinases. This chemical soup acts like an infectious rot. It degrades the extracellular matrix, breaks down collagen, weakens tissue elasticity, and signals neighboring healthy cells to also enter senescence. As we age, our immune system becomes sluggish and loses the ability to hunt down and destroy these rogue entities. They accumulate exponentially in our joints, leading to arthritis, in our blood vessels, causing cardiovascular stiffness, and in our brains, driving neurodegenerative decline. The human reality of cellular senescence is that aging is not a clean, uniform wear and tear of materials. It is an active, self-inflicted poisoning. We do not just rust out like old cars. We are actively corrupted from within by our own broken components that refuse to let go of life, turning our inner biology into a crowded graveyard of living ghosts. Picture the chromosomes inside your cells as long, intricate pieces of genetic thread. At the very ends of these threads sit tiny, protective plastic caps, identical to the aglets on the tips of your shoelaces. These caps are called telomeres. Every single time your cells divide to heal a wound, grow new skin, or keep your organs functioning, these plastic caps are violently shaved down, becoming shorter and shorter. Your telomeres are a literal, physical fuse burning down inside your body. The moment that fuse hits zero, the cell suffers absolute genetic collapse, its code unravels, and it loses the ability to regenerate, marking the definitive mathematical end of your biological youth. The discovery of telomeres and their relationship to aging earned Elizabeth Blackburn, Carol Greider, and Jack Szostak the Nobel Prize in 2009. Telomeres exist because of a fundamental glitch in how DNA replicates. The enzymes responsible for copying our genetic code cannot copy the very tip of the DNA strand. They require a baseline structural runway to sit on. As a result, with every single cycle of cellular division, a small segment of the telomere is left uncopied and lost forever. When telomeres become critically short, the cell interprets this as a broken, fragmented strand of DNA and locks down all operations, entering a permanent state of arrest. This attrition is the foundational clock of human mortality. But what makes telomere science deeply human is that the burning speed of this fuse is highly sensitive to psychological and emotional reality. Pioneering studies showed that mothers caring for chronically ill children, individuals enduring extreme, unyielding psychological stress, possessed telomeres that were significantly shorter than women of the exact same chronological age. The sheer weight of their emotional grief and cognitive anxiety was physically accelerating the degradation of their DNA, shaving off up to a decade of cellular life. This reveals an undeniable visceral connection between mind and matter. Our thoughts, our social safety networks, and our emotional traumas are not abstract, fleeting experiences. They are translated into physical forces that grind away at our genetic shields, dictating exactly how fast our biological fuse melts into the dark. Think about the beautiful, crispy, golden brown crust on a freshly baked loaf of bread, or the rich, seared surface of a perfectly cooked steak. In the culinary world, this delicious browning process is called the Maillard reaction, a chemical bonding between sugars and proteins under high heat. Now, realize that this exact same chemical reaction is happening inside your warm 98.6-degree body every single minute of your life, completely independent of a stove. Over decades, the simple sugars floating in your bloodstream are slowly, systematically bonding to the delicate proteins in your eyes, your skin, and your arteries, literally frying your tissues from the inside out, turning your flexible, youthful body into a stiff, brittle piece of biological caramel. This internal browning process results in the formation of advanced glycation end products, appropriately abbreviated as AGEs. When excess glucose or fructose molecules circulate through your cardiovascular system, they spontaneously collide with vital structural proteins like collagen and elastin. Without needing any enzymes to guide them, these sugars latch onto the amino acids, forming chaotic, irreversible crosslinks. Once these crosslinks are forged, the structural integrity of the protein is permanently ruined. Collagen, which should be supple and elastic to keep your skin smooth and your blood vessels pliable, becomes rigid, cross-linked, and stiff. This is the primary driver behind the formation of deep wrinkles, the loss of skin elasticity, and the dangerous hardening of the arteries that leads to strokes and heart attacks. Furthermore, ages bind to specific cellular receptors, RAGE, triggering a massive chronic wave of oxidative stress and systemic inflammation throughout the body. The human impact of glycation is particularly devastating because it represents a slow, silent crystallization of our inner architecture. When you consume a diet high in processed sugars and refined carbohydrates, you are not just gaining body fat. You are actively accelerating the internal caramelization of your tissues. Your kidneys lose their filtration capacity because their delicate membranes become clogged with glycated proteins. Your eyes develop cataracts as the lenses lose their transparency due to sugar-bound proteins. Ages prove that aging is quite literally a slow-cooking process, a chemical hardening of our flexible foundations that gradually transforms a vibrant, fluid organism into a fragile, unbending sculpture of sugar and ash. Imagine living in a massive, sprawling metropolis where the central power grids are slowly, systematically blowing out. One by one, the generators inside the power plants sputter, spark, and lose their capacity to produce electricity, plunging neighborhoods into total darkness. This catastrophic grid failure is happening right now across the vast cellular landscape of your body. The microscopic power plants inside your cells, the mitochondria, are decaying. As they break down, they stop producing the vital energetic currency that keeps your heart pumping, your brain thinking, and your muscles moving, leaving your body trapped in an unyielding state of systemic brownout that leaves you perpetually exhausted and vulnerable to disease. Mitochondria are the absolute engines of eukaryotic life. Billions of years ago, they were independent bacteria that entered an evolutionary pact with our ancient cellular ancestors. Today, they contain their own unique, highly vulnerable loop of DNA, mtDNA, and are responsible for generating adenosine triphosphate ATP, the universal fuel that powers every single biological action in the human body. However, the process of making ATP is incredibly violent. As mitochondria burn oxygen to create fuel, they constantly leak highly reactive, destructive molecules called free radicals. Because mitochondrial DNA lacks the robust protective shields and advanced repair mechanisms found in the main nucleus of the cell, it sits directly in the line of fire, taking constant, devastating hits from its own toxic exhaust. Over decades, this localized bombardment causes extensive mutations in the mitochondrial code. The damaged power plants begin to deform, losing their structural efficiency. They produce less and less ADP while leaking vastly greater amounts of oxidative stress into the cell. This energy crisis is the core reason why aging is characterized by a profound, inescapable drop in physical vitality. When your mitochondria decay, your brain cells lack the power required to process memory, leading to cognitive decline. Your cardiac muscles lack the energy to pump blood efficiently, and your metabolic systems slow to a crawl. The human experience of aging is ultimately an energetic bankruptcy. As our cellular generators burn themselves out from their own internal fires, the brilliant lights of our physical and mental capacities slowly fade, proving that life is entirely dependent on the preservation of our ancient, microscopic sparks. Imagine a silent, microscopic wildfire raging through your body. There are no sudden, agonizing pains, no swollen joints, and no visible rashes to alert you to the danger. Instead, a low-grade, toxic, and persistent chemical fire is quietly burning through your bloodstream, melting away the delicate lining of your arteries, dissolving the neural connections in your brain, and degrading the architecture of your vital organs. This is inflammaging. a devastating, chronic state of sterile inflammation that your body enters automatically as it grows older, transforming your own life-saving immune system into a weapon of mass destruction that slowly incinerates your youth from within. The term inflammaging was coined by Italian immunologist Claudio Franceschi in 2000 to describe a universal, inescapable feature of human aging. When we are young, inflammation is a beautiful, highly controlled emergency response. If you cut your finger or catch a virus, your immune system fires a precise, localized burst of inflammatory chemicals to destroy invaders and clear away damaged tissue. Once the battle is won, the system shuts down and peace is restored. But as we age, our immune cells become exhausted, confused, and hypersensitized. They lose their precision controls. The accumulation of cellular debris, zombie cells, and damaged DNA constantly triggers the immune system's alarm bells, even when there is no active infection. The body enters a permanent state of high alert. Macrophages and T cells begin circulating through your system, continuously pumping out destructive inflammatory cytokines like interleukin-6, IL-6, and tumor necrosis factor alpha, TNF-alpha. This is a sterile war. There is no foreign enemy to fight, so the immune system begins attacking your own healthy tissues. The human cost of this internal fire is immense. Inflammaging is now recognized as the common underlying engine driving almost every major age-related disease, from Alzheimer's and type 2 diabetes to osteoporosis and cardiovascular disease. It means that growing old is akin to living inside a house where the smoke alarms are permanently blaring and the sprinkler systems are continuously dumping acidic water on the furniture. Your body becomes trapped in its own defensive loop, destroying its foundation in a desperate, broken effort to protect itself from the ghost of decay. Deep within the dark, protected marrow of your bones and the hidden recesses of your organs sits a highly specialized elite force of biological shapeshifters. These are your adult stem cells. They are the ultimate internal construction crews, possessing the magical ability to transform themselves into any cell type your body needs to rebuild a shattered bone, replace a damaged liver, or smooth out a patch of aged skin. But as the decades roll by, a devastating shortage strikes your biological grid. Your stem cells lose their ability to replicate, their numbers plummet, and they enter a state of deep, irreversible exhaustion. Your body runs out of bricklayers, leaving you with empty toolboxes and a crumbling physical frame that can no longer repair its own daily fractures. Stem cell exhaustion is one of the definitive hallmarks of organismal aging. In our youth, our bodies are defined by a beautiful, near-infinite capacity for regeneration. If you damage a muscle or injure an organ, chemical distress signals immediately alert the local stem cell niches. These cells wake up, divide rapidly, and differentiate into fresh, pristine, and fully functional tissue cells, completely erasing the damage. However, stem cells are not immune to the wear and tear of time. They live under a strict biological budget. Every time a stem cell divides to save your life, it takes a hit to its telomeres and accumulates minor genetic errors. Over time, to protect the body from spreading mutated cells that could turn into cancer, the stem cells enter a state of forced retirement. They lose their potency and stop dividing. The consequences for human life are catastrophic. Without active stem cells, your skin thins and loses its ability to heal quickly from simple scratches. Your muscle mass steadily vanishes because your satellite stem cells can no longer repair the microtears caused by physical exertion. Your immune system decays because your hematopoietic stem cells can no longer manufacture a diverse army of fresh white blood cells. The human experience of aging is the tragedy of a builder who has lost the master blueprints and run out of raw materials. The structural damage of daily life accumulates completely unchecked, transforming a self-healing biological masterpiece into a fragile structure that can only watch its own gradual breakdown. Imagine living in a beautiful luxury apartment building where the waste management systems have completely broken down. The garbage collectors have gone on strike, and the trash chutes are permanently jammed. Day after day, you are forced to stack bags of rotting waste in your living room, your kitchen, and your hallways. Eventually, the trash fills the entire space, burying your furniture and making it completely impossible to walk, cook, or breathe. This exact garbage crisis is currently suffocating the longest-lived cells in your body. A stubborn, indestructible metabolic sludge called lipofuscin is packing into your heart muscles and brain cells, slowly choking out your vital internal machinery until the cell literally suffocates under the weight of its own undisposed waste. Often referred to by pathologists as the aging pigment, lipofuscin is a highly complex, yellowish-brown aggregate of oxidized lipids, cross-linked proteins, and heavy metals. It is the ultimate toxic byproduct of cellular life. Inside every cell sits an organelle called the lysosome, the cellular recycling plant filled with powerful enzymes designed to shred and dissolve broken components. Throughout your life, the lysosome works tirelessly to clean the cell. However, certain damaged proteins and fats become so warped and cross-linked by free radicals that the lysosomal enzymes cannot break them down. Instead of being recycled, these fragments fuse together into solid, sticky clumps of lipofuscin. Because the cell has no mechanism to eject solid waste through its outer membrane, the lipofuscin is permanently trapped inside. This accumulation is especially severe in postmitotic cells, cells that never divide, such as your cardiac myocytes and neurons in your brain. By the time you reach 80 years old, lipofuscin can take up to 30% of the internal volume of a heart muscle cell. This massive accumulation physically displaces the working organelles, disrupting the transport of nutrients, choking off energy production, and destabilizing cellular signaling. The human reality of lipofuscin is a visceral reminder of our structural limitations. Aging is not just an exhaustion of fuel or an unraveling of code. It is a slow, unyielding process of sedimentation, an internal pollution where we are progressively buried alive beneath the toxic, indestructible garbage of our own metabolism. Stand up and touch the skin on the back of your hand. Pull it gently upward, and notice how it instantly snaps back into place like a crisp, elastic rubber band. Now, imagine that over the next 40 years, an invisible, microscopic web of iron threads weaves its way through that skin, locking the fibers together in a rigid, frozen grid. The next time you pull that skin, it moves like stiff cardboard, refusing to stretch and staying tented in the air. This rigid freeze is happening to the collagen matrix that holds your entire body together, locking your joints, your skin, and your major organs inside a tight, unyielding biological straitjacket that robs you of your physical freedom. Collagen is the most abundant protein in the human body, serving as the primary structural scaffolding for our skin, bones, tendons, blood vessels, and internal organs. In our youth, collagen molecules are arranged in beautiful, organized, parallel bundles that can slide past one another smoothly, giving our tissues an incredible balance of strength and flexibility. However, as time passes, a destructive process known as non-enzymatic cross-linking takes over the matrix. Driven by oxidative stress, free radicals, and sugar molecules, stray chemical bonds begin forming horizontally between the parallel collagen strands. Think of it as turning a smooth, sliding ladder into a rigid, cross-hatched cage. These crosslinks permanently lock the collagen fibers into place, preventing them from moving or absorbing mechanical shock. The human experience of this structural freeze is what we commonly identify as the physical feeling of growing old. It is the reason why your joints feel incredibly stiff and creaky when you wake up in the morning. The tendons have lost their give. It is the reason your blood pressure spikes as you age. Your aorta has become so cross-linked and rigid that it can no longer expand to accommodate the pulsing rush of blood from your heart, forcing your cardiovascular system to work under dangerously high pressures. The cross-linking of collagen reveals that aging is a literal petrification, a slow chemical transition from a fluid, dynamic organism of motion into a brittle, locked structure that breaks instead of bends. Imagine waking up one day, looking in the mirror, and realizing that a quiet, invisible thief has been slicing away at the very foundations of your physical strength. Without you ever noticing, this thief has been stealing nearly 1% of your total muscle mass every single year. Your arms and legs are steadily thinning, replaced by soft, inert pockets of metabolic fat. The simple acts of lifting a grocery bag, climbing a flight of stairs, or even standing up from a deep, comfortable chair suddenly feel like monumental, exhausting athletic feats. You are entering the grip of sarcopenia, the universal, age-related wasting of human muscle flesh that slowly strips away your physical independence until you are left trapped inside a weak, fragile prison of your own bone. Sarcopenia, derived from the Greek words meaning poverty of flesh, is one of the most visible and debilitating aspects of human aging. Beginning around the age of 30, the human body undergoes a profound shift in its anabolic balance. As hormones like testosterone, growth hormone, and insulin-like growth factor, IGF-1, naturally decline, the body loses its drive to maintain costly muscle tissue. Concurrently, there is a selective, devastating loss of type 2 fast-twitch muscle fibers, the exact fibers responsible for generating explosive power, speed, and balance. This muscle wasting is compounded by neurogenic aging. The motor neurons in your spinal cord that connect your brain to your muscles begin to die off, leaving muscle fibers orphaned and disconnected from the neural grid. Without neural stimulation, these fibers atrophy and vanish, their space permanently colonized by fat and fibrous tissue. The human impact of sarcopenia is deeply tragic. It shifts a human being from a state of physical mastery to a state of profound vulnerability. It is the primary reason why falls become so deadly for the elderly. Without the fast-twitch muscle power to rapidly catch oneself during a slip, a simple loss of balance turns into a catastrophic, hip-shattering impact. Sarcopenia teaches us that our physical strength is a highly volatile, temporary loan. If we do not actively fight to preserve our flesh through resistance training and metabolic care, our biology will ruthlessly liquidate our muscles, leaving us structurally bankrupt long before our hearts stop beating. Nestled deep in your chest, directly behind your breastbone and sitting right above your heart, lies a small, butterfly-shaped organ called the thymus. In your childhood, this organ was a bustling, high-output military academy, working around the clock to train a specialized army of immune cells known as T cells to identify, track, and destroy lethal viruses and cancers. But as you hit puberty, a terrifying self-destruct sequence initiated inside this vital shield. The thymus began to rapidly shrink, shrivel, and dissolve, replacing its brilliant immune machinery with useless clumps of yellow fat. By the time you reach old age, your ultimate protective academy is virtually gone, leaving your body defenseless against mutated pathogens and silent, emerging tumors. This brutal process is known as thymic involution, and it is one of the most enigmatic and destructive ironies of human biology. The thymus hits its peak size and output during childhood, ensuring that our immune systems are thoroughly educated against the threats of the world. However, as sex hormones flood the body during adolescence, they trigger a rapid structural degeneration of the organ. The functional cortical and medullary tissues of the thymus are progressively replaced by adipocytes, fat cells, at a rate of roughly 1 to 3% per year. By age 60, the thymus is reduced to a vestigial shadow of its former self, producing a mere trickle of new, naive T cells. The consequences for human health are staggering. This collapse is the primary driver of immunosenescence, the age-related decay of the immune system. Because the body can no longer generate fresh, unprogrammed T cells, it must rely on an aging, exhausted army of memory T cells that have already been deployed against past infections. When a novel pathogen emerges, or when a brand new type of cancer cell mutates in your lungs or colon, the immune system lacks the flexible, freshly trained recruits needed to recognize the threat. This is why a simple bout of influenza or a respiratory virus that a child easily brushes off can be a swift, fatal death sentence for an older adult. Thymic involution reveals that our biological defense system is on a strict timer. We are gifted a powerful, youthful shield for only a brief window of our lives, after which our inner military academy closes its doors, leaving us to fight an increasingly hostile world with a fading veteran guard. Imagine having a twin brother or sister. You share the exact same DNA, the same sparkling eyes, and the same smile. When you are 5 years old, you are virtually indistinguishable. But as you both blow past your 50th birthdays, a strange divergence takes place. You look at each other and realize that your faces, your health profiles, and even your personalities have radically split. One twin is vibrant and running marathons, while the other is battling chronic illness and cognitive decline. Your identical genetic codes have drifted apart. An invisible, chaotic force called epigenetic drift has scrambled the software of your bodies, proving that even with identical blueprints, time will ruthlessly customize and warp our individual paths to the grave. Epigenetic drift describes the gradual, chaotic divergence of our epigenetic markers over the course of a lifetime. While our genome, the hard-coded DNA sequence, remains stable, the epigenome acts as the dynamic software layer that dictates which genes are turned on or off in specific cells. When twins are born, their epigenetic software is perfectly synchronized. Their genes are expressed in identical patterns. However, as they move through different lives, encountering unique mixtures of stress, sleep patterns, dietary choices, environmental toxins, and random molecular accidents, their epigenetic maps begin to accumulate tiny, uncoordinated mutations. Over decades, this creates a profound structural divergence. Genes that are supposed to protect the body against cancer can become accidentally silenced in one twin, while genes that drive chronic inflammation are mistakenly switched on. A famous study tracking identical twins across their lifespans visually demonstrated this drift. Young twins showed near-identical genomic methylation profiles, whereas older twins displayed massive, chaotic variations across their entire DNA landscape. The human truth of epigenetic drift is both liberating and terrifying. It shatters the myth of genetic determinism, proving that we are not entirely trapped by the cards we are dealt at birth. But it also exposes the cold reality of entropy. As we age, the software of our life becomes increasingly disorganized, losing its original harmony and drifting into a personalized, chaotic noise that slowly dissolves our physical identity. Inside the delicate, golden-lit neural pathways of your brain sits a vast population of tiny, specialized guardian cells called microglia. They are the ultimate internal doctors and security guards of your mind, constantly traveling between your neurons, cleaning up metabolic debris, and protecting your consciousness from infections. But as the years pass and your brain endures systemic stress, a dangerous mutation takes place within these guardians. They become primed. They morph into hyper-aggressive, paranoid entities that sit perfectly still, waiting for the slightest metabolic trigger to release a catastrophic, unchecked flood of neurotoxic chemicals that physically dissolves your synapses and sets your memory banks ablaze. Microglial priming is one of the most terrifying frontiers in modern neurogerontology. In a young, healthy brain, microglia exist in a highly active surveying state. They use long, elegant cellular branches to gently touch and monitor the health of neurons and synapses. If they encounter a threat, they temporarily shift into an inflammatory state to clear the danger, then quickly return to their peaceful baseline. However, as the brain ages, it is subjected to a constant barrage of low-grade systemic inflammation, oxidative stress, and microstrokes. This chronic exposure fundamentally breaks the microglia's regulatory systems, locking them into a state of permanent hyperreactivity known as priming. A primed microglial cell is a ticking time bomb. It looks peaceful on the surface, but its internal machinery is packed with inflammatory cytokines and destructive free radicals. The moment the body experiences a peripheral stressor, such as a common bladder infection, a bout of pneumonia, or even an intense period of psychological grief, the systemic inflammatory signals travel up into the brain. The primed microglia completely overreact, launching a massive, out-of-control chemical assault. Instead of protecting the neural grid, they begin systematically chewing through healthy synapses, destroying neural connections, and driving widespread neuronal death. This is the primary cellular mechanism behind the sudden, terrifying episodes of delirium and permanent cognitive drops that older adults often experience following routine surgeries or physical illnesses. Microglial priming reveals that the aging brain is not simply running out of memory space. It is actively consumed by its own overzealous guardians, turning our internal security system into the ultimate architect of our cognitive demise. Imagine living inside a magnificent stone cathedral where a silent, invisible construction crew is working every single night. The crew is split into two teams: the excavators, who chisel away old, microfractured stone, and the masons, who immediately follow behind with fresh mortar to rebuild the walls. For decades, the cathedral remains perfectly solid. But as you cross into your 50s, the masons suddenly slow down, drop their tools, and abandon the site, while the excavators keep digging at double speed. Year after year, they carve away the structural stone, leaving the columns hollowed out until the entire massive cathedral becomes as fragile as a pane of glass, ready to shatter from a single gentle step. This structural uncoupling is the precise mechanism behind osteoporosis, the age-related degeneration of human bone architecture. Human bone is not a dead, static rock. It is a highly dynamic, living tissue that is completely replaced every 10 years through a process called bone remodeling. This process relies on a flawless, balanced dance between two cell types: osteoclasts, the excavators that dissolve old bone, and osteoblasts, the masons that build fresh bone matrix. In our youth, these cells operate in perfect harmony, maintaining a dense, powerful skeletal scaffold. However, as we age, systemic shifts, especially the dramatic drop in estrogen during menopause for women and the steady decline of testosterone in men, destroy this cellular coordination. The osteoblasts become sluggish and short-lived, while the osteoclasts become hyperactivated. The balance is permanently broken. The osteoclasts carve deep cavities into the trabecular bone, but the exhausted osteoblasts fail to fill the gaps. The interior of your bones transforms from a dense, thick honeycomb into a thin, hollowed-out web of fragile threads. The human reality of this uncoupling is a silent, terrifying fragility. A person can feel completely healthy, vibrant, and strong, unaware that their skeletal scaffold has been hollowed out from within. It often takes a sudden, minor accident, such as leaning over to hug a grandchild or tripping on a carpet, to cause a catastrophic fracture of the hip or a compression collapse of the spine. Bone remodeling uncoupling teaches us that the core structures holding us upright are locked in a continuous battle against time, and when our biological builders strike, our very frames will quietly hollow themselves out into dust. Deep within every single cell of your body sits a magnificent, automated self-eating machine called autophagy. It is the ultimate internal recycling plant, continuously hunting down warped proteins, broken mitochondria, and cellular debris, dragging them into a burning acidic chamber to be broken down into raw amino acids to build a fresh, clean future. But as you blow past your middle age, this life-saving recycling machinery jams. The sensors fail, the transport vehicles break down, and the acidic chambers lose their fire. Your cells lose the ability to clean themselves, leaving your internal space to choke on a mounting, toxic mountain of old molecular garbage that paralyzes your vital organs and freezes your biological clock. The discovery of the mechanisms of autophagy was so monumental to human health that it was awarded the Nobel Prize in Physiology or Medicine in 2016, given to Yoshinori Osumi. Autophagy, which literally translates from Greek as self-eating, is the body's primary defense system against cellular clutter and metabolic stress. When a cell is fasting or enduring exercise, autophagy ramps up, clearing away damaged components to keep the cell pristine and functional. However, one of the most devastating aspects of the aging process is the systemic, widespread failure of this recycling network. As we grow older, the genetic pathways that regulate autophagy, such as the mTOR and AMPK pathways, become chronically deregulated. The cell loses its ability to form autophagosomes, the cellular trash bags used to wrap up waste. Furthermore, the lysosomes, which act as the incinerators, become accumulated with indestructible waste like lipofuscin, losing their internal acidity. As a result, the recycling process grinds to a halt. The cell becomes a toxic, clogged warehouse. In the brain, this autophagy failure allows altered proteins like beta amyloid and tau to aggregate unchecked, directly driving the formation of the plaques and tangles that destroy memory in Alzheimer's disease. In the muscles, it leaves broken, radical-leaking mitochondria to poison the tissue, driving weakness and decay. The human truth of autophagy failure is that aging is a fundamental breakdown in basic cleanliness. We do not just run out of life. We are slowly choked out by the toxic debris of our own past experiences, proving that to survive, our bodies must never stop the radical, continuous process of tearing themselves down to rebuild anew. Imagine if deep within your genetic code lay a hidden, dormant fountain of youth, a magical enzyme that possesses the power to rebuild your melting telomeres, restore your frayed DNA, and grant your cells complete, unyielding immortality. This enzyme actually exists. It is called telomerase. In the earliest stages of your life, when you were a developing embryo in the womb, this enzyme was wide awake, keeping your cells perfectly young and infinitely dividing. But the moment you were born, your body executed a cold, calculated genetic lockdown. It turned off the master switch for telomerase across almost every cell in your body, padlocking the fountain of youth and explicitly condemning you to a lifetime of steady, mathematical decay. Telomerase is a specialized ribonucleoprotein reverse transcriptase enzyme that elongates the repetitive DNA sequences at the terminal ends of chromosomes. By adding new TTAGGG repeats to the tips of telomeres, telomerase completely neutralizes the end replication problem, allowing cells to divide indefinitely without ever burning down their genetic fuses. While it remains active in our stem cells, germ cells, and unfortunately in 90% of human cancer cells, it is completely silenced in normal somatic cells, the cells that make up our skin, organs, and muscles. This silencing is a brilliant, yet tragic evolutionary trade-off known as antagonistic pleiotropy. In the chaotic, wild history of human evolution, the primary threat to a young organism's survival was runaway hyperproliferative cancer. To protect us from dying of tumors before we could reproduce, evolution chose to shut down telomerase in our everyday tissues, creating a strict limit on how many times a cell can divide. The cost of this cancer protection is the inevitability of aging. As our somatic cells divide to heal our bodies over decades, they steadily march toward their Hayflick limit, running out of telomere length because the fountain of youth has been intentionally locked away. The human reality of telomerase silencing is a profound lesson in biological compromise. Our mortality was not an accident or a design flaw. It was an active evolutionary choice, a structural sacrifice where our individual capacity for eternal youth was systematically traded away to ensure we lived just long enough to pass the torch of life to the next generation. Imagine stepping into a high-end manufacturing plant where thousands of precision robotic arms are folding delicate sheets of steel into highly complex, three-dimensional engine parts. If a single part is folded incorrectly by even a fraction of a millimeter, it becomes a useless, hazardous piece of metal that can jam the entire factory floor. Now, imagine that as the factory ages, the robotic arms lose their calibration. They begin outputting millions of misshapen, twisted, and mangled parts that clump together, grinding the assembly lines to a definitive screeching halt. This mechanical breakdown is happening to the proteostasis network inside your cells, transforming your beautifully folded, functional life proteins into a tangled, toxic mass of structural junk. Proteostasis, short for protein homeostasis, is the highly complex, interconnected network of cellular machinery responsible for the flawless synthesis, folding, trafficking, and degradation of proteins. Proteins are the literal workhorses of life, everything from the hemoglobin carrying oxygen in your blood to the receptors intercepting thoughts in your brain relies on a protein being folded into an incredibly specific three-dimensional shape. To maintain this flawless architecture, the cell deploys an army of molecular chaperones, heat shock proteins that act as quality control engineers, guiding nascent protein chains into their correct shapes and refolding those that warp under heat or stress. As we age, this exquisite proteostasis network suffers an absolute operational collapse. The production of molecular chaperones plummets, and the cellular sorting mechanisms become overwhelmed by oxidative stress. As a result, proteins begin misfolding at an exponential rate. Denatured, unfolded protein chains expose their sticky hydrophobic interiors, causing them to aggregate into dense, indestructible clumps. The human impact of this structural collapse is written across the ledger of all neurodegenerative diseases. In Parkinson's disease, misfolded alpha-synuclein proteins clump into toxic Lewy bodies that incinerate dopamine-producing neurons. In Huntington's and Alzheimer's, it is the accumulation of huntingtin and tau aggregates that destroys the neural grid. Proteostasis collapse reveals that life is an ongoing, energetic battle against physical entropy. The moment our molecular chaperones lose their ability to maintain the exquisite, twisted architecture of our proteins, our cellular foundations lose their function, melting down into a chaotic tangle of useless, broken code. Imagine living in a luxurious mansion surrounded by an absolute mountain of fresh food, clean water, and rich resources. Yet, every single time you walk into the kitchen, the smart home computer system screams an alarm through the speakers. Warning! Absolute starvation imminent. Shut down all heaters, ration all resources, and prepare for famine. Fooled by the false alarm, you sit shivering in the dark, destroying your own furniture for fuel while surrounded by abundance. This exact communication breakdown is happening between your diet and your cells. Your biological nutrient-sensing networks have become broken and deregulated, blinding your body to the resources around you and forcing your metabolism into a state of self-destructive panic. Deregulated nutrient sensing is a foundational hallmark of aging that alters how our bodies convert food into cellular energy and growth. Our cells rely on four major, highly evolutionary conserved nutrient-sensing pathways to read the environment: mTOR, which detects amino acids and drives growth; AMPK, which senses low energy and drives survival; sirtuins, which track NAD+ levels; and the insulin/IGF-1 signaling pathway, which responds to carbohydrates. In a young, healthy body, these pathways operate like a flawless thermostat. When food is abundant, insulin and mTOR fire up, signaling the body to build muscle, grow tissue, and replicate cells. When food is scarce, AMPK activates, shifting the body into a pristine state of maintenance, repair, and deep cellular cleaning. As we grow older, this delicate metabolic thermostat shatters. The pathways become chronically deregulated and desensitized. Even when we are overnourished, the cells fail to read the signals correctly. mTOR can become locked in a hyperactive state, continuously driving cellular growth and blocking the life-saving cleaning processes of autophagy, leading to obesity, insulin resistance, and cancer. Conversely, the body can become resistant to insulin, leaving glucose to rot in the bloodstream while the interior cells starve for energy. The human reality of deregulated nutrient sensing is that our modern lifestyle, characterized by continuous, unyielding access to high-calorie food, directly breaks the ancient sensory switches that kept us young. By constantly forcing our bodies to play the song of growth, we break the metabolic gears that handle repair, convincing our cells to burn through their limited life budget at a catastrophic pace. Imagine that inside your body, every single cell carries a tiny, invisible punch card hanging around its neck. Every time the cell divides to heal a scratch, replace a lining, or grow a tissue, a sharp mechanical punch clamps down, leaving a hole in the card. The card has exactly 50 slots, not 51, not 60. The very second that final 55th punch is made, a definitive biological lockdown occurs. The cell's machinery freezes, its access to life is revoked, and it enters an irreversible state of permanent retirement. This is the Hayflick limit, an absolute, unyielding molecular expiration date hard-coded into human tissue that ensures no matter how healthy your lifestyle is, your body will eventually run out of the ability to replicate itself. Before Leonard Hayflick's historic discovery in 1961, the scientific community believed a beautiful, seductive lie, that human cells were inherently immortal and that aging was simply a vague, systemic accumulation of body-wide wear and tear. The legendary Nobel laureate Alexis Carrel had claimed to keep chicken heart cells alive in a laboratory dish for over 30 years, leading everyone to believe that if you kept cells nourished, they would live forever. Hayflick proved this was a myth. In a series of elegant experiments, he demonstrated that normal human fetal fibroblasts grown in a pristine culture medium will divide exactly 40 to 60 times before hitting a hard wall of senescence. This finite replicative lifespan is a direct consequence of telomere shortening. The Hayflick limit is the biological realization of our mortality at the cellular scale. It means that our bodies are operating on a strict, pre-funded capital budget. Every single time your body is forced to heal itself from an injury, a toxic exposures, or chronic inflammation, it must spend a precious unit of its finite replicative capacity. When you smoke a cigarette and damage your lung lining, or sunbathe and incinerate your skin cells, you are forcing the remaining cells to divide at double speed to repair the gap. You are burning through your punch card at a rapid, reckless pace. The Hayflick limit is a humbling, scientific reminder that our physical existence has a strict, hard-coded border. We are built with an internal countdown timer, and every cellular renewal is a step closer to our ultimate expiration. Imagine sitting in a concert hall, listening to a world-class symphony orchestra play a breathtaking, perfectly synchronized piece of classical music. The violins, flutes, and horns are in absolute harmony. But as the performance goes on, a chaotic disruption takes place. Random audience members walk onto the stage, banging on drums, untuning the guitars, and screaming through megaphones. The beautiful symphony slowly dissolves into a horrific, deafening wall of pure static and noise. This sonic collapse is happening right now to the genetic symphony inside your organs, as random molecular static scrambles your epigenetic signals, transforming the clear music of your youth into a broken, unreadable mess of biological noise. While genetic mutations alter the actual letters of our DNA code, epigenetic noise describes something far more insidious: the gradual loss of transcriptional fidelity without any change to the underlying code itself. Every cell in your body contains the exact same dictionary of DNA, but a liver cell must only read the liver chapters, and a skin cell must only read the skin chapters. This precise reading is enforced by epigenetic marks that act as molecular bookmarks and highlights. In our youth, these marks are perfectly placed, ensuring that cells express their genes with absolute clarity and zero errors. However, as decades of environmental friction, thermal fluctuations, and free radical attacks hit the cell, these epigenetic bookmarks begin to slide, detach, and reattach in random, incorrect positions. This is the phenomenon of epigenetic noise. The cell loses its transcriptional discipline. The aging heart cell might accidentally begin expressing genes that belong to a bone cell. A kidney cell might start reading chapters intended for a stomach cell. The cell does not die, and it does not turn into cancer. Instead, it enters a state of deep functional confusion. It forgets its identity. The human experience of this molecular static is a systemic drop in organ efficiency. Your liver still looks like a liver on an ultrasound, but at the cellular level, its workers are reading a scrambled, noisy manual, executing their daily tasks with a staggering rate of errors. Epigenetic noise teaches us that aging is ultimately an informational catastrophe. We do not fade away because our blueprints vanish, but because the ink fades and the pages become so covered in chaotic scribbles that our bodies can no longer read the instructions for how to stay alive. I have prepared the first 20 facts, detailing them with structural depth, specific scientific mechanisms, and an engaging human tone. Imagine living in a magnificent, multi-story skyscraper where the invisible steel beams, concrete floors, and structural cables holding the rooms in place suddenly begin to soften and liquefy. The walls don't collapse all at once. Instead, the entire building begins to slowly sag, warp, and drift. The doors no longer fit their frames, the plumbing lines tear open, and the communication wires snap under the shifting weight. This structural liquefaction is happening right now within the spaces between your cells. Your extracellular matrix, the magnificent biological scaffolding that keeps your skin firm, your blood vessels intact, and your organs suspended in perfect geometric order, is melting away, turning your crisp, taut inner architecture into a collapsed fluid mess. The extracellular matrix, ECM, is not an inert biological jelly. It is a highly complex, dynamic, and non-cellular physical network made of collagen, elastin, laminins, and glycosaminoglycans like hyaluronic acid. The ECM acts as the physical environment in which your cells live, providing the mechanical signals that tell them how to behave, migrate, and survive. In your youth, the ECM is perfectly cross-linked and highly hydrated, acting like a pristine, tension-optimized trampoline. It ensures your skin bounces back instantly from a smile, your lungs expand effortlessly with air, and your arteries absorb the violent pressure waves of every heartbeat. As you age, this scaffolding faces a two-front assault. First, chronic low-grade inflammation upregulates a family of destructive enzymes called matrix metalloproteinases, MMPs. These cellular scissors relentlessly slice through the pristine networks of collagen and elastin. Second, the aging fibroblasts responsible for rebuilding the matrix go on strike, dramatically reducing their output of fresh structural proteins. The human consequence is a total breakdown in mechanical communication. When the ECM degrades, cells literally lose their footing. Deprived of physical tension, healthy cells enter apoptosis or turn senescent. The blood vessels lose their structural elasticity, causing them to stiffen and blow out under pressure. Skin collapses into deep, permanent folds because the underlying foundation can no longer support its weight. The degradation of the extracellular matrix teaches us that aging is not just a failure of cellular software. It is an absolute collapse of our physical architecture, a slow melting of the scaffolding that holds our very shape against the pulling force of gravity. Deep within the center of every single cell in your body sits the nucleus, the ultimate biological vault. This vault is wrapped in a highly fortified, dual-layer security membrane called the nuclear envelope, designed to keep your precious DNA locked away from the chaotic, chemical warfare raging in the outer cell. But as you age, a catastrophic structural failure strikes the vault walls. The security membrane begins to buckle, warp, and grow grotesque, blister-like bubbles called blebs. The protective gates snap open, allowing toxic cellular fluids to breach the core and directly corrupt your genetic master code, instantly aging the cell by decades and throwing its entire operational survival system into absolute chaos. The nuclear envelope is one of the most critical security boundaries in all of eukaryotic life. It is lined on its interior by the nuclear lamina, a dense, highly organized meshwork of intermediate filament proteins known as lamins, specifically lamin A, B, and C. This meshwork provides structural stiffness to the nucleus, organizes chromatin, and regulates gene expression. The most dramatic proof of the lamina's importance to human aging was discovered through the study of Hutchinson-Gilford progeria syndrome, a rare, devastating genetic disease where children age at 10 times the normal rate, dying of advanced heart disease by age 14. Progeria is caused by a single-point mutation that produces a deformed, toxic version of lamin A called progerin. Instead of forming a smooth, protective scaffold, progerin sticks to the nuclear envelope like toxic glue, causing the nucleus to lose its shape, develop severe blebbing, and fracture. What neurogerontologists have recently discovered is deeply humbling. Progerin is not exclusive to these sick children. Normal, healthy human beings actively produce small amounts of progerin as they grow older. Over decades, this toxic protein accumulates within our blood vessels, skin, and organs, slowly mimicking the exact same nuclear envelope disruption seen in progeria. As our nuclear walls buckle, the spatial organization of our DNA is shattered. Heterochromatin, the locked, silenced regions of our genome, unravels, allowing forbidden, ancient viral elements sleeping in our genetic code to wake up and trigger massive inflammatory responses. The disruption of the nuclear envelope proves that aging is a breach of our ultimate sanctuary. When the inner walls of our genetic vault fracture, the boundary between order and chaos dissolves, leaving our master blueprints to rot in the cellular wind. Imagine operating a hyper-advanced nuclear power plant where the spent, cracked, and radiation-leaking uranium fuel rods are supposed to be automatically removed by robotic arms and dropped into a thick containment cooling pool. Now, imagine that the robotic arms freeze. The cracked fuel rods remain locked inside the active reactors, heating up to volatile levels, cracking open, and spewing deadly, glowing radioactive waste directly onto the control room floor. This precise power plant meltdown is occurring across the energetic terrain of your body right now. Your mitophagy system, the specialized cleanup crew that hunts down and destroys broken, radical-spewing mitochondria, has completely collapsed, leaving your cells packed with volatile, toxic reactors that are actively incinerating your tissue from within. Mitophagy is a highly specialized form of selective autophagy that targets damaged or dysfunctional mitochondria for destruction. Because mitochondria are the primary generators of adenosine triphosphate, ATP, they are constantly subjected to immense oxidative stress, causing them to break down frequently. To prevent these damaged organelles from poisoning the cell, a beautiful genetic surveillance system involving two proteins, PINK1 and Parkin, acts as a cellular quality control unit. When a mitochondrion drops its voltage, PINK1 accumulates on its outer membrane and recruits Parkin, which coats the broken reactor in molecular destroy tags, signaling the autophagosome to swallow it whole. As we age, the genetic expression of PINK1 and Parkin drops significantly, leading to an absolute operational failure of mitophagy. The consequences for human health are devastating, particularly within the central nervous system. The brain consumes roughly 20% of the body's total energy budget, making it completely dependent on pristine mitochondrial health. When mitophagy fails, neurons become packed with deformed, swollen mitochondria that can no longer produce ATP. Instead of generating energy, these zombie reactors leak massive waves of reactive oxygen species, ROS, and open their permeability transition pores, dumping cytochrome C directly into the cytoplasm. This triggers the cellular suicide cascade, driving widespread, irreversible neurodegeneration. The collapse of mitophagy is the primary driver behind Parkinson's disease, where dopamine-producing neurons are systematically wiped out by mitochondrial toxicity. It reveals that our physical vitality is tied to a ruthless recycling schedule. The moment we lose the ability to clear away our broken inner reactors, they transform from the engines of life into the ultimate architects of our death. Imagine a master typist sitting at a desk, tasked with copying a massive 3 billion letter manual that contains the absolute instructions for how to keep you alive. In your youth, the typist is flawlessly accurate, typing with perfect speed and zero mistakes. But as the hours turn into decades, the typist grows profoundly exhausted. Their eyes blur, their fingers slip, and they begin making catastrophic typos on every single page. They delete entire paragraphs, repeat sentences indefinitely, and substitute vital words with gibberish. This exhausted typist is the replication machinery of your cells. Every single day, your DNA accumulates tens of thousands of structural fractures and typos, systematically destroying your genetic master manual until the instructions for life become completely unreadable. Genomic instability is widely considered the foundational first hallmark of aging. Our DNA is subjected to a relentless, unyielding bombardment of damage from both internal and external sources. Every single day, the DNA inside a single human cell takes up to 70,000 individual hits from ultraviolet solar radiation, environmental toxins, dietary mutagens, and the reactive oxygen species generated by our own metabolism. This damage manifests as single-strand breaks, highly lethal double-strand breaks, and chemical crosslinks. To counter this daily apocalypse, our cells have evolved a breathtakingly complex network of surveillance and repair enzymes known as the DNA damage response, DDR. Enzymes like ATM, ATR, and the famous tumor suppressor protein P53 constantly patrol the double helix, snipping out errors, pasting incorrect bases, and sealing broken strands. However, as time passes, the genes encoding these repair enzymes accumulate their own mutations. The repair machinery loses its efficiency. Unrepaired DNA damage accumulates exponentially across your tissues. When these genetic typos occur within vital homeostatic genes, the cell loses its ability to function. It can no longer manufacture essential enzymes, structural proteins, or metabolic receptors. If the damage hits a critical checkpoint, the cell either self-destructs via apoptosis, turns into a toxic senescent zombie, or breaks completely free from biological control to become a runaway cancerous tumor. The human reality of genomic instability is that our bodies are written in a fading ink. Every day we live, our genetic code becomes a little more corrupted, a little more fragmented, until the master manual that built us from a single cell is reduced to a chaotic, unreadable script of biological decay. Imagine living in a massive, modern country where the entire telecommunications network suddenly becomes infected by a malicious, destabilizing virus. The emergency hotlines are jammed with fake alerts. The military bases receive conflicting attack orders, and the civilian internet is flooded with toxic, terrifying propaganda that drives the public into an absolute state of mass panic and civil war. This systemic network breakdown is happening across the vast cellular civilization. Your intercellular communication channels have become completely corrupted. The chemical, hormonal, and electrical signals that allow your organs to work in perfect harmony have devolved into a toxic, chaotic wall of noise, forcing your tissues to actively attack and destroy one another. A human being is not an isolated collection of independent organs. We are a massive, highly integrated civilization of over 30 trillion cells that must constantly speak to one another to maintain life. This conversation is mediated through a highly complex array of circulating molecules. Endocrine hormones traveling through the blood, neurocrine signals jumping across synapses, and paracrine cytokines communicating between neighboring cells. One of the most destructive aspects of aging is the profound alteration and degradation of these communication networks. This hallmark is tightly linked to inflammaging and the presence of senescent cells. As zombie cells accumulate in tissues, their senescence-associated secretory phenotype, SASP, floods the local environment with highly inflammatory signaling molecules. This chemical pollution scrambles the normal, healthy dialogue between cells. Neuroendocrine pathways, such as the hypothalamic pituitary adrenal, HPA, axis, become chronically desensitized and hyperactivated, constantly pumping out stress hormones like cortisol that degrade muscle tissue and suppress immunity. At the same time, the blood-brain barrier breaks down, allowing inflammatory signals from the gut or liver to leak into the central nervous system, disrupting neural communication and driving cognitive decline. The human experience of this altered communication is a total loss of physiological resilience. Your body loses its ability to coordinate its defenses. The pancreas speaks, but the liver cannot hear. The immune system sees a minor threat but launches a catastrophic, self-destructive counterattack. It proves that aging is ultimately a crisis of community. We do not collapse because our individual cells fail, but because the vital bonds of communication that bind them into a single, unified life dissolve into a deafening wall of pure, chaotic noise. Deep within every cell of your body sits a collection of small, highly fortified bubbles called lysosomes. These are your cellular stomach chambers, packed with an incredibly volatile, burning mix of highly concentrated digestive acids and flesh-melting enzymes designed to shred waste. To keep the cell safe, these acidic chambers are sealed behind a specialized, heavy-duty membrane shield. But as the decades roll by, a terrifying structural disaster occurs. Oxidative stress punches millions of microscopic holes through the lysosomal walls. The protective shields fail, and the burning, flesh-melting digestive acids leak directly out into the delicate interior of the cell, literally digesting your cellular components from the inside out in a slow, agonizing chemical meltdown. Lysosomal membrane permeabilization, LMP, is a critical, highly destructive cellular event that accelerates the kinetics of aging and tissue degeneration. The lysosome is the ultimate recycling hub of the cell, maintaining an internal pH of roughly 4.5 to 5.0, vastly more acidic than the surrounding cytoplasm. This intense acidity is required to activate over 50 different types of hydrolytic enzymes, known as cathepsins, which can rapidly break down proteins, lipids, and old organelles. As we age, the lysosomal membrane is subjected to a constant, unyielding assault from free radicals and the accumulation of undigestible metabolic waste like lipofuscin. Lipofuscin acts like a physical abrasive, destabilizing the lipid bilayer of the lysosome. When the membrane integrity is compromised, cathepsins leak out into the neutral cytoplasm. If the leak is massive, it triggers a swift form of programmed cell death known as lysosomal apoptosis. However, if the leak is low-grade and chronic, it induces a state of permanent sublethal damage. The leaked digestive enzymes slowly degrade vital functional proteins, break down structural networks, and damage neighboring organelles without killing the cell outright. This chronic acid leak plays a massive role in degenerative diseases like osteoarthritis, where leaked lysosomal enzymes escape the chondrocyte and systematically dissolve the protective cartilage matrix of your joints. It is also a primary driver of muscle wasting and macular degeneration in the eyes. Lysosomal membrane permeabilization reveals that our cells are operating directly on top of volatile chemical vaults. Aging is the slow, unyielding corrosion of the containment walls, a silent disaster where the very tools designed to clean and preserve our life dissolve their shields and become the ultimate instruments of our internal destruction. Imagine stepping into a world-class television broadcasting studio where the master computers are completely taken over by a silent, invisible software virus. The virus doesn't delete the film files or smash the cameras. Instead, it subtly alters the transmission codes. It dials the volume of important news broadcasts down to an unhearable whisper, while turning the volume of pure static and emergency alarms up to a deafening shriek. The audience is left utterly confused, unable to decipher what is real. This exact software sabotage is happening to your genetic expression right now. A hidden family of microscopic genetic controllers called microRNAs has become completely dysregulated, silencing your body's life-saving survival codes while blasting the signals of decay. MicroRNAs, miRNAs, are a class of small, non-coding RNA molecules, typically only 19 to 25 nucleotides in length, that function as the ultimate post-transcriptional regulators of gene expression. They do not code for proteins themselves. Instead, they act like precision genetic volume knobs, binding to specific messenger RNA, mRNA, strands to either block their translation or mark them for immediate destruction. A single microRNA can control the expression of hundreds of different genes simultaneously, making them the master software architects of cellular identity and metabolic health. One of the most complex frontiers in longevity science is the discovery that human aging is characterized by a sweeping, systemic dysregulation of the microRNA profile, a phenomenon known as miRNA drift. As we age, the expression of specific, life-preserving microRNAs like the miR29 and miR34 families changes dramatically. For instance, the downregulation of miR146A allows the chronic inflammatory fire of inflammagen to burn completely out of control across your cardiovascular system. At the same time, spikes in other malicious microRNAs systematically silence the expression of sirtuins and DNA repair enzymes, effectively paralyzing the cell's ability to handle stress. The human impact of this software glitch is profound. It explains why aging bodies suddenly lose their metabolic flexibility. The genetic volume knobs have been twisted out of calibration by time. Your cells still possess the hard-coded blueprints to heal, build muscle, and burn fat, but the microRNA software layer has dialed those essential instructions down to an unreadable whisper, forcing your biology to play a chaotic, distorted loop of systemic vulnerability. Imagine your DNA as a massive, epic library containing thousands of ancient, locked books that hold the secret spells for absolute health, youth, and cellular resilience. In your youth, these books are kept open and accessible because they are located near the protective ends of your chromosomes. But as your telomeres shorten with age, an eerie, spatial transformation occurs. The chromosomes physically fold and buckle in on themselves, dragging those vital open books deep into the dark, suffocating center of the genetic pile. The books are slammed shut and permanently padlocked, silencing your body's ultimate survival codes simply because the geometric geography of your DNA has shifted. The telomere position effect, TPE, and its age-related variant TPEOLD, telomere position effect over long distances, is a mind-bending epigenetic phenomenon that links the physical length of telomeres directly to the structural expression of distant genes. For decades, scientists believed that telomeres were purely passive caps that only affected the cell when they ran out completely at the Hayflick limit. TPOLD shattered this assumption, proving that telomeres act as dynamic, long-range spatial anchors for the entire genome. Chromosomes are not straight lines. They are looped into complex, three-dimensional shapes inside the nucleus. When telomeres are long and youthful, they create specific loops that keep certain genes located millions of base pairs away in an open, active architectural zone known as euchromatin. One classic example is the HTERT gene, which codes for telomerase itself, and the ISG15 gene, vital for antiviral immunity. As telomeres progressively shorten across your lifespan, the physical anchor point slips. The loop structures lose their tension and collapse. The distant survival genes are dragged into dense, tightly packed silencing zones known as heterochromatin. The cell physically loses its ability to read these genes, completely independent of any mutations to the code itself. The human reality of TPOLD is a striking display of biological geometry. It means that long before a cell hits its absolute replication limit and dies, the progressive shortening of its telomeric caps is actively rewriting its genetic geography, systematically locking away the books of youth and forcing the cell to operate on a highly restricted, heavily censored script of survival. Deep within the cytoplasm of your cells floats an array of tiny, specialized chemical processing plants called peroxisomes. Their job is highly dangerous. They break down toxic fatty acids and neutralize the lethal hydrogen peroxide generated by your metabolism using specialized enzymes to strip the volatile elements away. But as you age, these chemical plants suffer an absolute containment failure. The entry gates warp, the processing enzymes are locked outside, and the peroxisomes transform from life-saving waste treatment plants into highly volatile oxygen bombs that leak pure, unadulterated chemical acid directly into your cellular matrix, vaporizing your internal structures. Peroxisomes are essential, single membrane-bound organelles involved in lipid metabolism and the regulation of cellular oxidative stress. They contain high concentrations of catalase, a hyper-efficient enzyme capable of converting millions of molecules of toxic hydrogen peroxide \BMH_20_2 into harmless water and oxygen every single second. Peroxisomes are also responsible for alpha and beta oxidation of very long-chain fatty acids, VLCFAs, which are vital for maintaining the myelin sheaths that insulate the nerves in your brain. As we age, a critical transport system known as the peroxisomal targeting signal (PTS) pathway breaks down. Cells lose the ability to import freshly manufactured catalase into the interior of the peroxisomes. As a result, peroxisomes become empty, non-functional ghosts, while catalase sits uselessly in the cytoplasm. Deprived of their protective enzymes, peroxisomes can no longer neutralize the massive waves of hydrogen peroxide generated during fatty acid metabolism. The internal pressure blows out the organelle's structural integrity, causing it to leak highly reactive oxygen radicals across the cell. This peroxisomal degeneration has catastrophic consequences for human physiology. Without functional peroxisomes, very long-chain fatty acids accumulate to toxic levels in the blood, directly attacking the white matter of the brain, destroying the myelin insulation, and accelerating the onset of neurodegenerative decline and cognitive slowing. Peroxisome degeneration proves that our cells are locked in a continuous chemical balancing act. When the precision gates of our internal waste treatment plants warp, the very chemicals meant to be safely neutralized are unleashed as a toxic flood, burning down the structural foundations of our health. Imagine stepping into a high-stakes emergency room where the waste disposal bins are completely overflowing with used syringes, bloody bandages, and toxic biohazard waste. The doctors can no longer reach the operating tables because the floor is buried under a mountain of medical garbage. This exact operational paralysis is crippling your aging cells. Your proteolysis systems, the specialized molecular machines tasked with shredding and clearing away old, damaged proteins have become completely paralyzed. Your cells have transformed into stagnant, crowded protein graveyards where your vital cellular machinery is physically choked out by a mounting, uncleared mountain of molecular debris. Altered proteolysis is a core component of the broader hallmark known as loss of proteostasis. To maintain cell survival, the life cycle of a protein must be strictly regulated from birth to death. When a protein has fulfilled its purpose or becomes damaged by oxidative stress, it must be rapidly degraded through two primary proteolytic pathways: the ubiquitin proteasome system, UPS, and the lysosomal autophagy system. The UPS acts like a precision molecular paper shredder, where damaged proteins are tagged with a chain of small molecules called ubiquitin, which acts like a postal zip code directing them into the core of the 26S proteasome barrel to be sliced into tiny peptides. As we move through the lifespan, this precision shredding operation suffers a total mechanical failure. The enzymatic activity of the proteasome barrel plummets, and the ubiquitin ligases lose their tracking accuracy, often tagging healthy proteins while ignoring highly toxic aggregates. Compounding the crisis, heavily cross-linked protein clumps, such as those formed by advanced glycation end products, AGEs, physically plug the entry pores of the proteasome barrel, completely jamming the shredder mechanics. The human cost of this proteolytic paralysis is devastatingly vast. In the muscle cells, undegraded structural proteins accumulate, disrupting sarcomere alignment and accelerating sarcopenia. In the cardiovascular system, altered proteolysis prevents the clearance of damaged endothelial proteins, driving the stiffening of the vessel walls. Altered proteolysis teaches us that life requires a relentless, highly efficient system of destruction. When our internal shredders jam, our cells lose their ability to clear the past, turning our internal architecture into a stagnant graveyard of broken tools that smothers our biological future. Deep within the dark, heavily guarded epicenter of your cell's nucleus sits a small, dense, hyperactive structure called the nucleolus. This is the ultimate engine of cellular manufacturing, the factory that constructs ribosomes, which are the specialized molecular machines responsible for assembling every single protein that keeps your heart beating and your brain thinking. But as you grow old, the nucleolus encounters a devastating operational crisis. The ribosomal DNA templates warp, the assembly lines fracture, and the factory floor enters an absolute shutdown. Your body enters a catastrophic ribosome famine, losing its ability to manufacture the very workers needed to repair your fading flesh. The nucleolus is the absolute command center of protein translation machinery. It is responsible for the transcription and processing of ribosomal RNA, rRNA, and the assembly of ribosomal subunits. Because the nucleolus must maintain an incredibly high metabolic output, transcribing thousands of copies of rRNA continuously, its DNA templates, rDNA, are highly repetitive and uniquely vulnerable to homologous recombination errors, double-strand breaks, and transcription-induced stress. One of the newest and most exciting frontiers in longevity science is the discovery that the nucleolus acts as a central hub for the aging process. As we age, the repetitive rDNA architecture experiences progressive stability loss. The constant transcriptional friction causes the nucleolus to fragment, lose its structural boundaries, and enter a state of chronic nuclear stress. When this occurs, ribosome biogenesis drops to near zero levels. Deprived of fresh ribosomes, the cell can no longer execute protein translation efficiently. This ribosome famine triggers a powerful checkpoint response. The free ribosomal proteins leak out into the nucleoplasm, where they bind to and inhibit MDM2, a protein that normally suppresses the tumor suppressor p53. This stable activation of p53 forces the cell to permanently cease division and turn senescent. The human reality of nuclear stress is an absolute manufacturing freeze. Your cells can have perfect hard-coded blueprints and plenty of raw amino acids, But if the internal factories that build the manufacturing machinery itself break down, your body can no longer field the workers required to maintain your life, forcing your tissues to slowly stall and fade into structural bankruptcy. Imagine a highly classified military base where the most dangerous, unstable weapon designs and ancient, chaotic war secrets are locked away inside high-security underground vaults, sealed behind concrete walls and heavy steel doors. Now, imagine that as the base grows old, the concrete walls slowly crumble into sand and the steel doors rust off their hinges. The dangerous secrets and volatile, forbidden blueprints leak out onto the public streets, causing absolute, terrifying chaos across the nation. This security breach is happening inside your DNA right now. Your heterochromatin, the highly compressed, locked vaults of your genetic code, is disintegrating with age, allowing forbidden, destructive genes to break completely free from control. The human genome is broadly divided into two structural states: euchromatin, loose, active, and easily read DNA, and heterochromatin, tightly packed, highly condensed, and transcriptionally silenced DNA. Heterochromatin is vital for maintaining genomic stability, as it keeps repetitive DNA sequences, transposable elements, and ancient retroviral sequences securely locked down so they cannot replicate and disrupt our functional genes. This tight compaction is maintained by specific epigenetic marks, such as histone H3 lysine 9 trimethylation, H3K9Me3, and the presence of structural proteins like heterochromatin protein 1, HP1. One of the most universal characteristics of organismal aging is the global systemic loss of heterochromatin, a phenomenon known as heterochromatin loss. As we age, the levels of H3K9Me3 and HP1 drop significantly, causing the tightly packed vaults of our DNA to uncoil, unravel, and relax. The consequences for human biology are insidious. When heterochromatin dissolves, ancient transposable elements called line 1 retrotransposons, which make up nearly 17% of the human genome and have slept since the dawn of evolution, suddenly wake up. They begin cutting themselves out of the DNA and pasting themselves into random, functional gene sequences, causing widespread genomic typos and double-strand breaks. Furthermore, this loose, uncontrolled gene expression forces the cell to waste immense metabolic energy transcribing forbidden sequences, directly triggering a powerful type I interferon immune response that drives the burning fire of inflammaging. The depletion of heterochromatin reveals that our youth depends entirely on what we keep locked away. When our genetic vaults lose their structural tension, the chaotic ghosts of our evolutionary past break free to tear down our cellular present. Right now, floating through your bloodstream is a massive, trillion-strong army of highly trained killer white blood cells known as T-cell. Enter a state of deep, absolute exhaustion. They drop their weapons, freeze mid-track, and develop specialized molecular breaks that lock them in a state of permanent paralysis, leaving your body completely exposed to the rapid rise of lethal tumors. T-cell exhaustion is a distinct, highly problematic state of immune dysfunction that characterizes the aging immune landscape, contributing heavily to the phenomenon of immunosenescence. In a young body, when a T-cell encounters an antigen from a virus or a mutated tumor, it activates aggressively, secretes destructive cytokines like interferon gamma, and rapidly proliferates to wipe out the threat. Once the infection is cleared, the army retracts, leaving behind a highly efficient squad of memory cells. However, as we age, our bodies are subjected to decades of persistent, low-grade viral stimulation from chronic pathogens like cytomegalovirus, CMV, which infects up to 80% of the adult population. This unyielding, decades-long antigen exposure fundamentally breaks the T-cell's internal wiring. To prevent the immune army from causing catastrophic autoimmune damage to the body through continuous firing, the T-cells activate a primitive feedback loop, expressing inhibitory receptors on their surface known as immune checkpoints, specifically PD1, CTLA4, and TIM3. These checkpoint receptors act like heavy mechanical brakes. When a tumor cell or a novel respiratory virus encounters an exhausted T cell, it simply taps these surface receptors, instantly paralyzing the immune cell. The exhausted T cell loses its ability to proliferate, drops its cytokine output, and can no longer kill target cells. The human reality of this immune paralysis is the sudden, exponential spike in cancer rates that characterizes old age. Your body does not run out of white blood cells. Instead, your internal army becomes a field of paralyzed, exhausted veterans, standing completely frozen in the shadow of the invading enemy. Imagine a magnificent, lush oasis packed with billions of vibrant, green plants, all kept alive by an incredibly intricate network of millions of microscopic irrigation pipes that deliver fresh water to every single square millimeter of soil. Now, imagine that as the oasis ages, a silent disaster occurs under the dirt. One by one, the microscopic pipes begin to crack, clog, and completely dissolve. The irrigation grid steadily vanishes. Within decades, the outer edges of the oasis dry up, turning into a barren, cracked, and lifeless desert terrain where the cells literally wither away from absolute dehydration and starvation. Microvascular rarefaction is the age-related, progressive loss of the microscopic blood vessel networks, specifically the capillaries that feed our tissues. Every organ in the human body is completely dependent on capillary density. These tiny vessels, often only a single cell layer thick, are the exclusive sites where oxygen, glucose, amino acids, and vital hormones are exchanged between the blood and our living tissue cells. As we age, our tissues undergo a sweeping drop in the expression of angiogenic factors, most notably vascular endothelial growth factor, VEGF. Without steady VEGF signaling, the endothelial cells that form the walls of our capillaries lose their survival signals and undergo apoptosis. The microvascular grid begins to systematically prune back, a process highly accelerated by sedentary lifestyles and metabolic dysfunction. The human consequences of microvascular rarefaction are profoundly devastating. In the skeletal muscles, the loss of capillaries limits oxygen delivery, directly driving the muscle wasting of sarcopenia and leaving you perpetually fatigued from minor physical exertion. In the skin, rarefaction causes the dermal layers to thin and go cold, losing their ability to regulate temperature. Most critically, microvascular rarefaction in the brain slashes cerebral blood flow, starving neurons of glucose and oxygen, driving the white matter hyperintensities seen on neuroimaging and accelerating vascular dementia. Rarefaction teaches us that aging is a slow process of structural desertification. As our invisible, microscopic pipelines of life wither and vanish, our tissues are left stranded in a biological drought, slowly starving in the dark long before our central pulse gives out. Imagine walking into a high-stakes factory where the aging workers are feeling incredibly weak, exhausted, and slow. To compensate for their lack of speed, the workers do something highly grotesque. They begin eating continuously, blowing up to 3 times their normal physical size. They become massive, swollen giants that take up the entire factory floor, crushing the delicate machinery around them and consuming all the power grid's electricity, while outputting a mere fraction of the actual work. This distorted expansion is happening inside your aging organs right now, as your cells undergo a destructive, swollen mutation known as senescent hypertrophy. While we often think of aging as a process of simple atrophy and shrinkage, at the cellular level, the opposite is frequently true. When cells enter a state of senescent arrest, or when postmitotic cells like cardiac myocytes and neurons endure chronic metabolic stress, they activate the mechanistic target of rapamycin (mTOR) pathway in an unyielding, unregulated loop. mTOR is the primary engine of cellular mass growth, driving the rapid translation of proteins and accumulation of lipids. In our youth, mTOR activation is highly controlled, allowing our tissues to grow to their correct genetic boundaries. But as we age and lose our nutrient-sensing discipline, the mTOR pathway becomes chronically locked in the on position. Because senescent cells have permanently lost the ability to divide, they cannot split their volume through mitosis. Instead, they just keep growing larger and larger, expanding into grotesque, hyperfunctional shapes. This cellular hypertrophy causes immense physical and metabolic strain. The swollen cells demand a massive share of the body's glucose and oxygen, starving healthy neighboring cells. In the heart, cardiac myocyte hypertrophy forces the walls of the ventricles to become incredibly thick and rigid, preventing the heart chambers from relaxing between beats, a primary cause of diastolic heart failure in older adults. In the kidneys, podocyte hypertrophy stretches the filtration barriers, causing vital proteins to leak out into the urine. The hypertrophy of aging reveals a profound paradox. Our decay is not driven by a simple running down of our inner gears, but by an unbridled, toxic overactivation of growth, a swollen momentum where our cells expand into monstrous shadows of themselves until they crush the very systems that sustain them. Imagine a magnificent, towering rope ladder stretching miles into the sky, holding the structural integrity of your life together. Every single day, a malicious saboteur walks up to the ladder with a pair of heavy steel shears, snipping through individual ropes. A single cut is easily repaired. But eventually, the saboteur clamps down on both sides of the ladder simultaneously, snapping the entire structure completely in half. The ladder breaks, the pieces twist into chaos, and everything crashing down into absolute ruin. This catastrophic fracture is happening to your DNA double helix right now. As you age, double-strand breaks accumulate unchecked across your code, cleanly snapping the genetic ladder of your life in two. Among all the various forms of DNA damage that drive genomic instability, the DNA double-strand break, DSB, is considered the absolute most lethal and mutagenic. A single-strand break is easily fixed because the opposing strand acts as a perfect template for repair. But a double-strand break represents a clean, total bisection of the double helix. The physical continuity of the chromosome is lost, leaving two fragmented, unanchored ends floating blindly inside the nucleus. To repair this catastrophe, cells deploy two primary pathways: homologous recombination, HR, a high-fidelity, perfect repair mechanism that operates during cell division, and non-homologous end joining, NHEJ, a sloppy, emergency glue job that simply pastes the two nearest broken ends together, frequently causing massive deletions and insertions of random base pairs. As we grow older, the high-fidelity HR pathway fails completely, forcing the cell to rely exclusively on the error-prone NHEJ mechanism. Over decades, this sloppy emergency patching leaves our chromosomes heavily scarred, rearranged, and structurally unstable. Chromosomal translocations run rampant. When a DSB occurs within an essential housekeeping gene, the damage triggers the immediate activation of the ATMP53 pathway, forcing the cell to either execute apoptosis or enter the toxic zombie state of senescence. The human impact of DSB accumulation is a progressive, structural unraveling of our genetic identity. It is the primary mechanism behind the rapid, age-related degeneration of highly active tissues like the intestinal lining, the skin, and the blood vessels, proving that our life is built upon a delicate, twisting ladder that time systematically shears into fragmented, unreconstructible dust. Deep inside a single one of your healthy cells sits a thriving population of up to several thousand independent mitochondria, each carrying its own unique, ancient loops of circular DNA. In your youth, this genetic ecosystem is perfectly uniform. Every single loop contains an identical, pristine code for making energy. But as the decades roll by, a silent, microscopic civil war erupts within the cell. Mutated, deformed loops of mitochondrial DNA begin to replicate at double speed, actively outcompeting and overtaking the healthy genes. Within decades, your cells enter a state of genetic chaos called heteroplasmy, transforming your internal energy grid into a broken, warring battlefield that can no longer produce the fuel of life. Mitochondrial DNA, mtDNA, is uniquely vulnerable to mutation because it sits directly next to the electron transport chain, where highly destructive reactive oxygen species are continuously generated as a byproduct of ATP production. Furthermore, mtDNA lacks the histone shielding and advanced nucleotide excision repair pathways that protect the main nuclear genome. When a mutation occurs in an mtDNA loop, it typically damages a gene responsible for a vital respiratory chain protein. The mind-bending nightmare of mitochondrial biology is that mitochondria replicate independently of the cell cycle through a process of random clonal expansion. If a mutated mtDNA loop loses its structural efficiency but retains its replication anchors, it can actually replicate faster than healthy loops or evade the cell's mitophagy cleanup cruise. This state, where a single cell contains a mixed population of both healthy and mutated mtDNA, is known as heteroplasmy. As long as the percentage of mutated mtDNA remains low, below 60%, the cell can function normally. But once the heteroplasmy threshold is crossed, an absolute energetic catastrophe strikes. The mutated loops dominate the ecosystem. The cell's respiratory chains break completely down, ADP output crashes to near zero, and the cell is flooded with massive waves of oxidative stress. The human reality of heteroplasmy is a patchy, highly erratic decay of our most energy-hungry tissues. It explains why aging muscles experience sudden, localized fiber death, and why the aging brain develops random, scattered pockets of metabolic failure, proving that our ultimate decline is driven by a microscopic evolutionary war, an internal civil conflict where our broken energetic codes out-replicate the structures that keep us alive. Imagine having a massive, priceless spool of exquisite silk thread that stretches for over 6 feet. To keep the thread from tangling into an unreconstructible, knotted ball of junk, you wind it perfectly around hundreds of tiny, polished wooden bobbins. As long as the bobbins are there, you can smoothly unravel and read the silk whenever you want. But now, imagine that as time passes, the bobbins begin to crack, splinter, and completely vanish. The 6-foot silk thread drops into a chaotic pile on the floor, knotting itself into a tight, tangled mess that can never be unraveled or read again. This exact structural disaster is striking your DNA right now, as your cell loses its histone chaperones, turning your organized genome into a tangled, knotted ball of genetic static. If you uncoiled the DNA inside a single one of your microscopic cells, it would stretch to a physical length of roughly 2 meters. To pack this massive thread into a nucleus that is only a few micrometers wide, the double helix is wrapped tightly around specialized, spool-like protein complexes called histones. This highly organized DNA protein architecture is known as chromatin. The precise winding, unwinding, and positioning of these histone spools is managed by an elite class of molecular workers called histone chaperones, such as ASF1 and FACT. One of the most profound, low-level hallmarks of aging is the progressive loss and degradation of these histone chaperones. As we grow old, the genetic expression of these chaperone proteins plummets. Without molecular chaperones to guide them, histones become unstable and detach from the DNA strand. The entire structural integrity of chromatin collapses. The 2-meter genetic thread drops into a disorganized, tangled state. Regions of the DNA that are supposed to be kept strictly wound up and silenced suddenly uncoil, exposing forbidden genes and ancient viral sequences to random transcription. At the same time, regions containing vital survival codes become knotted and buried, making them completely inaccessible to the cell's manufacturing enzymes. The human impact of this structural collapse is a total loss of transcriptional discipline. The cell loses its ability to rapidly adapt to stress, such as heat, toxins, or viral infections, because its genetic filing system has become a tangled, knotted warehouse, proving that our biological youth depends entirely on the polished spools that keep our massive threads of life in perfect, unyielding order. Imagine living in a vast, peaceful empire where the cities communicate entirely by sending millions of sealed, high-security mail trucks across the highways every single day. The trucks carry vital architectural blueprints, medical supplies, and coordination plans. But as the empire falls into decay, a rogue, hostile territory takes over the postal hubs. They intercept the mail trucks, emptying out the medicine and filling the cargo bays with highly toxic chemical bombs, explosive instructions, and infectious viruses, sending them out to detonating across every unsuspecting city in the land. This biological postal hijacking is happening inside your body right now, as your extracellular vesicles transform into poisoned letters that spread the sickness of aging from cell to cell. Extracellular vesicles, EVs, which include exosomes and microvesicles, are tiny, membrane-bound nanoshuttles secreted by cells to facilitate long-distance intercellular communication. These biological capsules travel through the bloodstream, saliva, and cerebrospinal fluid, packed with a highly specific cargo of microRNAs, functional proteins, and signaling lipids. When an EV reaches a target cell, it fuses with its membrane, dumping its cargo directly into the interior to alter its behavior. One of the most startling discoveries in modern longevity science is that aging causes a profound, destructive decoupling of the EV communication network. In our youth, EVs secreted by stem cells travel through the body, delivering regenerative signals that heal tissues, suppress inflammation, and maintain metabolic flexibility. However, as our tissue cells turn senescent and enter the zombie state, they hijack the body's postal system. Senescent cells begin hypersecreting a unique, highly toxic class of EVs known as senescent exosomes. These toxic capsules are packed with inflammatory microRNAs and destructive proteins from the SASP chemical soup. As they circulate through your blood vessels, they deliver a potent, infectious payload of aging to perfectly healthy, distant organs. A healthy young cell that swallows a senescent exosome can be instantly forced into senescence itself, spreading the rot of decay across your biological grid. This EV decoupling explains why aging accelerates so rapidly once it takes hold. Our internal communication network has been systematically weaponized against us, transforming our life-saving nanoshuttles into poisoned letters that broadcast the mandate of decay to every corner of our flesh. Imagine a highly advanced, ultra-wealthy nation where the central banks suddenly execute a catastrophic operational error. They stop printing money. Day after day, the currency circulating in the economy systematically evaporates. Within a few short years, the banks run completely dry, businesses close their doors, factories stop production, and the entire civilization grinds to an absolute, freezing halt simply because there is no longer a currency to facilitate the transaction of life. This economic collapse is devastating the cellular landscape of your body right now. Your systemic levels of NAD+, the ultimate molecular currency required for every single energetic and survival transaction in your flesh, are plummeting toward absolute bankruptcy. Nicotinamide adenine dinucleotide, \BM, NAD+, is one of the most essential coenzymes for life, present in every single living cell across all kingdoms of nature. \BM, NAD+ plays a dual, completely irreplaceable role in human biology. First, it acts as a vital electron shuttle in the metabolic reactions that convert glucose and fat into ATP inside our mitochondria. Second, and most critically for aging, \BM, NAD plus acts as the mandatory fuel for two massive families of defense enzymes. Sirtuins, the master longevity regulators that manage epigenetic silencing and DNA repair, and PARPs, the DNA damage response teams that patch double-strand breaks. The tragedy of human biology is that as we age, our systemic /BM, NAD plus levels undergo a catastrophic exponential collapse. By the time you reach age 50, your /BM, NAD plus reserves are slashed by half compared to your youth. By age 80, they drop to a mere fraction of that. This depletion occurs because of a devastating double whammy. Our bodies produce less /BM, NAD plus through the salvage pathways, while an aggressive, age-related enzyme called CD38 hyperproliferates across our immune cells, ruthlessly consuming and vaporizing our remaining /BM, NAD plus reserves like a molecular Pac-Man. The human impact of this currency crisis is an absolute failure of our biological defense systems. Without /BM, NAD plus, your sirtuins fall completely asleep. They can no longer maintain heterochromatin compaction or coordinate DNA repair, leaving your genome to unravel into chaos. Your PRP enzymes freeze mid-track, unable to seal double-strand breaks. The collapse of systemic /BM, NAD plus levels means that your cells are structurally starving in a land of plenty, entering an absolute state of energetic bankruptcy where they can no longer afford the basic cost of staying alive, forcing your entire biological empire to systematically freeze and fade into the dark. Imagine a highly fortified military bunker where the commanding generals hold the grand strategic blueprints for survival, while down in the basement, the engineers manage the raging furnace that keeps the lights on. To survive an enemy siege, these two rooms must talk constantly. Now, imagine a malicious spy cuts the intercom wire. The generals above cannot hear that the furnace is overheating, and the engineers below have no idea what the defense plan is. They begin firing blind, throwing random switches, and blasting toxic heat until the entire bunker explodes from within. This fatal miscommunication is tearing your cells apart right now. The vital communication wire between your master cell nucleus and your mitochondrial power plants has been cleanly severed by time. For a cell to function optimally, its two independent genomes, the 20,000 genes inside the cell nucleus and the 13 essential genes hidden inside the mitochondria, must maintain a flawless, real-time conversation known as mitonuclear communication. This dialogue is heavily mediated by the coenzyme /BM, NAD plus. When /BM, NAD plus levels are high and youthful, a master transcription factor called HIF1 alpha is kept strictly locked down, allowing a beautiful, smooth stream of protein signals to flow back and forth between the two compartments. As we age and systemic /BM, NAD plus levels plummet, this molecular intercom shatters, triggering a devastating state of pseudohypoxia. The nucleus can no longer read the state of the mitochondria, mistakenly believing that the cell is starving of oxygen. In a blind panic, the nucleus shuts down the transport of critical structural components that the mitochondria need to maintain their electron transport chains. Deprived of nuclear support, the mitochondrial proteins begin to mismatch. The structural subunits of the respiratory chain become highly unstable and misaligned. The energetic consequences for human biology are immediate and devastating. The mitochondria lose their capacity to generate ATP efficiently, while leaking a massive, destructive torrent of free radicals into the cell. This broken telephone is the core reason why aging tissues cannot adapt to sudden metabolic demands, such as a sprint, a cold winter night, or a sudden illness. Your organs possess the structural components to survive, but because the master control room and the engine room can no longer hear each other's voices, the entire metabolic grid enters an uncoordinated, chaotic brownout that stalls your vitality. Picture the most magnificent highway network in the world, stretching for over 60,000 miles inside your body. To ensure blood flows smoothly at supersonic speeds, the entire interior wall of this highway is lined with an ultra-slick, microscopic layer of living Teflon, just a single cell thick. This precious lining is your endothelium. In your youth, it is perfectly frictionless, dynamically expanding and contracting to guide life-giving oxygen to every organ. But as you pass your prime, a silent catastrophe occurs. This biological Teflon strips away, transforming your smooth, glassy pipelines into a rough, sticky, and heavily scarred battlefield where rogue fats and clotting cells smash into the walls, trapping your entire body inside a ticking cardiovascular time bomb. The endothelium is not a passive plastic pipe. It is the largest endocrine organ in the human body, responsible for synthesizing nitric oxide /BMNO, the master molecule of cardiovascular life. Nitric oxide acts as a powerful chemical messenger, commanding the smooth muscle cells surrounding your arteries to relax, allowing blood to flow effortlessly while preventing blood clots and cholesterol plaques from sticking to the vessel walls. As we move through the lifespan, chronic exposure to low-grade inflammation, circulating sugars, and free radicals damages the endothelial cells, a state known as endothelial dysfunction. The enzyme responsible for making /BMNO, endothelial nitric oxide synthase, ENOS, becomes uncoupled. Instead of generating life-saving nitric oxide, it begins pumping out superoxide radicals, actively destroying its own structural lining. The human impact of this shift is catastrophic. Without /BMNO, your blood vessels lose their ability to dilate. They become permanently stiff, narrow, and rigid. This chronic contraction forces your heart to pump blood against an immense wall of resistance, driving systemic hypertension and leading directly to strokes and heart attacks. Furthermore, the rough, unlubricated endothelial lining allows low-density lipoprotein, LDL, cholesterol to tear through the surface layer, initiating the massive calcified arterial plaques that characterize cardiovascular decay. Endothelial dysfunction reveals that our age is truly defined by the state of our pipelines. When the invisible, single-cell shield of Teflon grinds down into rust, our entire circulatory tree hardens, choking off the very currents that keep our flesh alive. Imagine a malicious arsonist creeping through your house, leaving tiny, sticky drops of combustible chemical oil on your carpets and curtains. By themselves, the drops are bad enough, ruining the look of your home. But the real horror begins when these sticky chemical oils collide with highly specialized security sensors on your walls. The moment they touch, the sensors malfunction, interpreting the sticky drops as an absolute military attack. The smart home system responds by deploying automatic flamethrowers that set your entire living room completely ablaze. This self-inflicted firestorm is precisely what happens inside your tissues when advanced glycation end products plug into the terrifying cellular receptors known as RAGE. Advanced glycation end products, AGEs, are the caramelized clumps of sugar and protein that stiffen our tissues as we age. While their physical presence ruins structural flexibility, their true pathogenic power is unlocked when they bind to a specific surface receptor appropriately named RAGE, receptor for advanced glycation end products. When an AGE molecule docks into a RAGE receptor on a cell surface, it acts like a master key that unlocks a massive inflammatory Pandora's box. The binding event triggers a continuous signal cascade that activates nuclear factor kappa B, NF-BM/kappa B, the master genetic switch for inflammation. Once NF-BM/kappa B is unleashed, it commands the cell's nucleus to continuously manufacture and secrete a devastating cocktail of inflammatory cytokines, including interleukin-1, IL-1, interleukin-6, IL-6, and tumor necrosis factor alpha, TNF-BM/alpha. This activation creates a highly destructive, self-perpetuating feedback loop. The inflammation generated by rage creates more oxidative stress, which accelerates the formation of more AGEs, which then plug into more rage receptors. The human cost of this vicious cycle is vast. It drives the rapid destruction of kidney filters in diabetic patients, burns through the delicate neural pathways of the brain, and systematically incinerates the collagen matrix of the skin. The age-rage axis proves that aging is not a passive decay. It is an active, amplified chemical firestorm where our own protective receptors are hijacked by metabolic waste, transforming a slow structural stiffness into a blazing, body-wide war of self-destruction. Imagine a high-security diamond cutting facility where millions of microscopic shards of razor-sharp glass dust are generated every hour. To keep the workers safe, the facility employs an elite squad of specialty janitors. These aren't normal cleaners with brooms. They are master engineers who inspect every single piece of dust, catch the most hazardous shards with precision tweezers, and drop them into a secure, heavy-duty chemical incinerator. Now, imagine these elite janitors walk out on a permanent strike. The razor-sharp glass dust accumulates across the floor, slicing through the workers' shoes, cutting the electrical lines, and bringing the entire high-tech operation to a bloody, grinding halt. This precise factory failure is crippling your cells as your chaperone-mediated autophagy undergoes an absolute, age-related decay. While macroautophagy handles large cellular structures like whole mitochondria, chaperone-mediated autophagy, CMA, is the cell's precision surgical strike team for individual damaged proteins. CMA relies on a highly specialized molecular chaperone called HSC70. This chaperone patrols the cellular interior, looking for a specific 5-amino acid barcode sequence, CFERC, exposed on proteins that have become warped or oxidized by free radicals. Once HSC70 hooks onto this barcode, it drags the damaged protein directly to the lysosome wall, where it binds to a specialized docking port called LAMP2A. The protein is then uncoiled and fed through the tight LAMP2A pore into the acidic interior to be shredded. The tragedy of aging is that the abundance of the LAMP2A docking ports on our lysosomal membranes plummets exponentially as we grow older. Without these ports, the entire CMA operation enters an absolute blockade. The HSC70 chaperones can still find the damaged proteins and tag them with precision, but when they drag them to the lysosome, the entry doors are completely boarded up. The elite janitors are locked out. As a result, highly toxic, misfolded protein monomers are left to float freely inside the cytoplasm. They bump into one another, cross-link, and form the devastating amyloid plaques and neurofibrillary tangles that define neurodegenerative diseases like Alzheimer's and Parkinson's. The decay of chaperone-mediated autophagy demonstrates that our cells are entirely dependent on their ultimate precision sanitation engineers. When our molecular entry doors rust shut, our inner space transforms from a high-tech masterpiece of life into a cluttered, glass-strewn floor that cuts down our survival. If these calm explorations help you unwind, you might also like, subscribe, or leave a thought below. It helps other sleepy souls find their way here, too. For now, let your thoughts settle and let the night carry you the rest of the way. Sleep well and good night.